De-extinction, or resurrection biology, or species revivalism is the process of creating an organism, which is either a member of, or resembles an extinct species, or breeding population of such organisms. Cloning is the most widely proposed method, although selective breeding has also been proposed. Similar techniques have been applied to endangered species. There is significant controversy over de-extinction, and critics assert that efforts would be better spent conserving existing species, and that the habitat necessary for formerly extinct species to survive is too limited to warrant de-extinction. Methods. Cloning is one method discussed as an option for bringing extinct species back. Proponents include author Stuart Brand, and proposed species include the passenger pigeon and the woolly mammoth. De-extinction efforts are now underway to revive the passenger pigeon by extracting DNA fragments and taking skin samples from preserved specimens, and, later, using band-tailed pigeons or rock pigeons as surrogate parents. A team of Russian and South Korean scientists, as of April 2013, in the planning stages to clone a woolly mammoth using an Asian elephant as a surrogate mother. Large amounts of well-preserved mammoth tissue have been found in Siberia. Once the process is completed, there are plans to introduce the mammoths to Pleistocene Park, a wildlife reserve in Siberia. Although de-extinction efforts have not yet succeeded in producing viable offspring of a previously extinct species, the same process has been applied successfully to endangered species. The banteng is the second endangered species to be successfully cloned, and the first to survive for more than a week. Scientists at Advanced Cell Technology in Worcester, Massachusetts, United States extracted DNA from banteng cells kept in the San Diego Zoo's frozen zoo facility, and transferred it into eggs from domestic cattle, a process called somatic cell nuclear transfer. 30 hybrid embryos were created and sent to Transova Genetics, which implanted the fertilized eggs in domestic cattle. 2 were carried to term and delivered by cesarean section. The first hybrid was born on April 1, 2003, and the second 2 days later. The second was euthanized, but the first survived and, as of September 2006, remained in good health at the San Diego Zoo. Scientists from the University of Newcastle and the University of New South Wales reported in May 2013 the successful cloning of the extinct frog, Rheabatrachus using the process of somatic cell nuclear transfer. The embryos developed for several days but died. In an important development, the scientists from Newcastle reported associated technologies that provide a proof of concept for the proposal that frozen zoos are an effective mechanism to provide an insurance against species extinction and the loss of population genetic diversity. They connected the circle between de-extinction and the prevention of extinction for threatened animal species. The important advances were the capacity to successfully recover live frozen embryonic cells from animals that produce large yolky eggs. When this development is combined with somatic cell nuclear transfer, it enables the genome to be recovered. The scientists point out that many embryonic cells can be frozen, and when combined with frozen sperm storage enables the genetic diversity of populations to be stored. With groups of vertebrates such as the amphibians facing an extinction crisis, they propose this as an effective means to prevent extinction while the causes of declines can be identified and remedied. The technical difference between frozen tissue samples commonly used for genetic studies and those in a frozen zoo is the use of cryoprotectants and special freezing rates at the time of freezing and thawing. Selective breeding, the aurochs, which became extinct in 1627, could possibly be brought back by taking DNA samples from bone and teeth fragments in museums in order to obtain genetic material to recreate its DNA. Researchers would then compare the DNA to that of modern European cattle to determine which breeds still carry the creature's genes, and then undertake a selective breeding program to reverse the evolutionary process. The intention would be that with every passing generation, the cattle would more closely resemble the ancient aurochs. Opposition. Opponents of de-extinction have claimed that efforts and resources to resurrect extinct species could have been better used trying to conserve endangered species that might themselves go extinct. It has also been noted that a resurrected species, while being genetically the same as previously living specimens, will not have the same behavior as its predecessors. The first animal to be brought back will be raised by parents of a different species, not the one that died out, and thus have differing mothering techniques and other behaviors. Scientific American, in an editorial condemning de-extinction, pointed out that the technologies involved could have secondary applications, specifically to help species on the verge of extinction regain their genetic diversity. For example, the black-footed ferret or the northern white rhinoceros. It noted, however, that such research should be conducted under the mantle of preserving modern biodiversity rather than conjuring extinct species from the grave. Other scholars have published ethical concerns regarding de-extinction. In conservation biology, Robert Sandler argues that introducing extinct species to environments may produce harm to modern species, as invasive species. Issues regarding scientific hubris, human and animal health, and the ecology of sensitive environments have been raised by the scientific community. Further research must be performed regarding de-extinction to investigate advantages and disadvantages to the technology. New technological practices must be examined to prevent environmental hazards.